So let's go to the second part. Okay, in the second part, I'm going to give you an overview about different human brain mapping methods that we are going to discuss through the session today, just to give you an idea about how they are related to each other. In this class that we are going to have today, there are different articles that we are using their kind of figures. I'm really interested to thank those people who have done really nice jobs in terms of kind of providing these educational resources and scientific evidence for us. It's also important for me to just kind of clearly explain to you that we are using the book that is called Fundamentals of Cognitive Neuroscience, and I kind of told you that the second author, Bernard Bars, is a famous person in the field of consciousness. So you can see that there are pieces in this kind of book that are mainly about consciousness. And the other author, Nicole Gage, Dr. Gage, she is mainly interested to kind of language and auditory processes. So you can see many pieces of evidence today that are related to language processing or auditory processing and auditory cortex and how we hear different sounds. So that is the reason that we have many pieces of evidence about that specific kind of area. And also, she's interested to kind of what we call MEG. I will discuss about those things. And you can see that there are different pieces of evidence about MEG. I try to kind of tailor the evidences that we are going to discuss about today in a way that would be sort of more balanced. And obviously, when you have an author, if I write a book, I definitely have kind of areas that I'm interested or areas that I'm more expert inside. And I will discuss about those evidences probably in a kind of higher detail. So that's what I was interested to just give you an idea about. Okay, there are some specific emphasis in this class about auditory systems. As you can see here in these kind of references, we have different references about auditory cortex, and there are some other kind of methods, papers that would be interesting for you. We have a major journal in the field that's called Neuroimage, and Neuroimage had a special issue in 2012, something like 8 years ago, specifically discussing about these different brain mapping methods. And that in that specific issue, there are several papers that are overviewing what is happening in the field, and you might be interested to see those papers. So that's the reason that we have at least 3 of those papers, one on EEG, one on MEG, the other on fMRI, as the main kind of resources for this book chapter or the session that we are going to have today. Okay, so to discuss about different methods of human brain mapping, what is critical for us, first of all, to have an idea about 2 major dimensions that are defining these 2, basically defining different brain mapping methods. For example, when we talk about fMRI, with fMRI, we cannot see neurons. So we do not have that resolution to get to the neuron level. We cannot even see what we call cortical layers. We might be able to see with some kind of trade-offs, some kind of things related to the column. So we might see things related to column. So that is probably the lower level of resolution, spatial resolution that we can get. But we cannot go any layer down from this side. And also what you can see here with fMRI, we have the temporal resolution. This is basically the temporal resolution. This one is spatial resolution. We are around the seconds and minutes, so we are around here when we talk about fMRI signal. So it's really hard to kind of talk about things that are happening in milliseconds or a portion of a second. So what we have in fMRI is mainly between few seconds to few minutes. So that is the temporal resolution of these two dimensions. And we have these dimensions in the log format because they are just going orders of magnitude. So it is not, when we go from millisecond to second to minute, just the orders of magnitude. So that's the reason that they use the term of log here, instead of just saying that it is just the time or the size. Let me show you one sample fMRI example, or sample fMRI kind of study. So you can see here that we have information every 2 seconds. So what we have here, we have the sampling rate of 2 seconds. And sampling rate in fMRI is called TR, or time to repetition. So when we see an fMRI experiment, so we can go and see what is the sampling rate. So sampling rate here is 0.4 hertz, 5 hertz. So that is the sampling rate, because we sample every 2 seconds. Okay. You can change the TR in the MR machine that you have. It's something like a kind of a control knob that you can kind of increase the TR or decrease the TR. But most of the studies are around 2, 3, 1 second. So that is the sampling rate that we can have. So the sampling rate is about 2 seconds. So we cannot really differentiate things that are happening under that resolution. So we cannot talk about things that are happening in the portions of the seconds. Okay. I'm not going to discuss about what does it mean and how, I mean these information is going to be helpful in the field, but I was just interested to give you an idea about, okay, the sampling is every 2 seconds here in an fMRI study. The other important issue about fMRI is that when we give people a stimuli here, there is always a lag between the stimuli that we give people and the peak of the signal that we have. And this lag is something like 6 seconds. So there is always a lag between the stimulus and the response that we receive. And the type of signal that we have is called bold, and it represents blood oxygenation. Blood oxygenation level dependent. So we are talking about oxygen. So it is not actually the neural activity. It is just the changes in the level of oxygen that we have around those neurons. So it is a sort of indirect measure of brain or neural activity. So we cannot say that we are recording neural activities. We are recording changes of oxygenations that are highly related to the neuronal activities. The other thing about fMRI is the amount of signal that we have in fMRI is something like 1–2%. So whatever we have in terms of the signal, if the signal is 100, when we do the stimulation, the signal would be summed with 2% of additional signal. So we need to be really careful to be able to detect just 2% of signal change. And this is not easy to do. So we need to be really kind of careful in terms of not to mess up noise with the real signal. And to have higher level of signal, most of the time we just give people multiple stimuli. So we give them repeated stimuli in a way to be able to have higher level of signal, up to 3–4% of the signal. So that is what we do usually in the field of fMRI, trying to give people a block of, this is what we call a block. We usually give them a block of a stimuli to get sure that we'll have enough signal to be able to detect. So when we kind of present people with a block of a stimuli, then we probably have enough signal to be able to detect. And there is a kind of accumulation of those signals together and give us something like a plateau of signal. Okay, that is another important thing that we need to know about what we call fMRI. The other important issue that we need to be careful and know about that is just this picture. If I just zoom up in this area in frontal cortex, this is what we are collecting in MRI. So these are basically a structural MRI, okay? this is what we see with what we call histology. So we can see the cells and whatever it is happening. This is 1 millimeter cubical. And if I zoom this up, these are the neurons that you can see. There are thousands of neurons here. Okay? So you can see thousands of neurons here. This is the smallest size that we can detect with MRI. Most of the MRI measures, most of the MRI kind of studies, we use what we call a voxel resolution of 1 millimeter cubical. So whatever we have in terms of the signal, we just make an average of what is happening in this 1 millimeter cubical of neurons, and we record that as one parameter or one measure. And we do that every 2 seconds. So whatever we have in terms of the data from the scanner is just giving us one value from each voxel, which are, at the best case scenario, 1 millimeter by 1 millimeter by 1 millimeter. Then they include something like thousands of neurons. We just make an average about what is happening in this voxel. And when we have the average, we just make one single value and report that every 2 seconds. So the data that we have with fMRI, or most of the kind of MR-based imaging methods over the time, is just a signal every 2 seconds from each of these voxels. Okay, so that is the resolution that we have. That is the spatial and temporal resolution. So to have an understanding about each of these brain mapping methods, we need to clearly know about the spatial and temporal resolution and what are the details that we can get into with these methods. Okay? When we talk about EEG, obviously what you can see here is the temporal resolution is much better. Why? Because in EEG we do 50 hertz, 100 hertz. There are machines that are doing sampling in something like 200, 300 hertz. And that means that we are doing the sampling 100 times every second. So the sampling rate is way higher. So if you want to buy an EEG machine, one of the things that you need to see is what is the sampling rate. The higher sampling rate, it means that you are in a really higher temporal resolution. So you can detect things that are happening in the milliseconds. Okay? But the challenge with EEG is that the spatial resolution is way worse than an fMRI. So it's really hard to see things that are in the maps and columns and kind of areas in the brain. So it just doesn't give us enough spatial resolution. Okay? So you can see that there are trade-offs between spatial and temporal resolution and different methods that we are using in the field. And then we have another method which is called PET, which has almost kind of really close spatial resolution to fMRI, but the temporal resolution is really bad. So whatever you are recording is about something like an average of 1 hour activity in the brain. So we make a picture from the brain about the activity that's happening in several minutes or up to something like an hour of activities. And we will discuss about PET as well. But the good thing about PET is that it can have a specific molecular targets. Something that it is really hard to be done with other methods. So for example, we can just do imaging for dopamine. We can just do the imaging about some specific chemicals in the brain. That would make things much, that makes PET something interesting to do. So you can see that each of these methods would have their own limitations and capacities. And then we have methods that are not only mapping, but also interventional. So we have interventional methods that would give us also mapping information. They also have a specific temporal and spatial resolution. Okay? When we talk about TMS, we can record things related even to up to something like up to tenths of seconds. We can because the TMS pulse is something like 1 millisecond. So we can have a good temporal resolution here with TMS, and we can go to up to something like the precision of up to, let's say, 5 millimeters. So it is not as good as fMRI probably to some extent, but the temporal resolution is better. So how these things are working, that's another thing to discuss, and we will discuss about that. And there are many other methods. So it's not just fMRI, PET, TMS, EEG. There are some other methods that we'll discuss about that might be interesting in the field. But these things that we have here, things like multi-unit recording, these are invasive methods. It means that we need to open the skull, we need to do surgeries, and these are the challenges that we are going to face if we want to use these methods. Okay.